Hello, teacher and classmates. Jimena and I will be talking about DNA. First, we're going to talk about characteristics of DNA. DNA has a unique double helix shape, as shown as in the image, like a twisted ladder. DNA is a two-stranded molecule. DNA contains four basic building blocks, or bases: adenine, thymine, cytosine, and guanine, as shown in the image. Your genome is made of a chemical called deoxyribonucleic acid, or DNA for short. Each base pair is joined by hydrogen bonds.
DNA is quite fragile, so errors during transcription aren't unusual. One of the most common ways it can be damaged is from ultraviolet light. Hopefully, there are many ways it repairs itself, so a lot of it can be fixed. Whatever isn't, though, becomes a mutation that we carry with us. The human genome consists of about 3 billion DNA base pairs. Between 5 to 8% of DNA is inhuman but from previous viruses. If we were to put all DNA molecules found in a human body from end to end, it could stretch from the Earth to the Sun and back over 600 times.
So, what is a nucleotide? A nucleotide consists of a sugar molecule, either ribose in RNA or deoxyribose in DNA, attached to a phosphate group and a nitrogen containing base. The bases used in DNA, as mentioned before, are adenine, cytosine, guanine, and thymine.
Where is DNA found in the cell? As we can see in the picture, DNA is located inside the cell's nucleus.
So, what's the process that occurs in cell reproduction in DNA? During mitosis, as shown in the image, a cell duplicates all of its contents, including its chromosomes, and splits to form two identical daughter cells. Meiosis also allows genetic variation through a process of DNA shuffling, while the cells are dividing. Mitosis and meiosis, the two types of cell division.
Now we will be talking about the discovery of DNA. In the year 1869, Friedrich Miescher identified the nuclein by isolating a molecule from a cell nucleus that would later become known as DNA. This was later followed by the German biochemist Albrecht Kossel identifying nuclein as nucleic acid. He also isolated those five nitrogen bases that are now considered to be the basic building blocks of DNA as well as RNA. In 1882, shortly after Kossel's findings, Walter Fleming became the first biologist to execute a wholly systematic study of the division of chromosomes. His observations that chromosomes double is significant to the theory of inheritance. During the early 1900s, Fyodor Boveri and Walter Sutton came up with the Boveri-Sutton chromosome theory. Their findings became fundamental in our understanding of how chromosomes carry genetic material and pass it down from one generation to the next one. In 1902, Mendel's theories were finally associated with a human disease by Sir Archibald Edward Garrett, who published the first findings from a study on recessive inheritance in human beings. In 1904, Oswald Avery first outlined DNA as the transforming principle, which essentially means that it's DNA, not proteins, that transform cell properties. This was followed by Chargaff discovering that DNA is responsible for heredity and that it actually varies between species. His discoveries, known as Chargaff's rules, proved that guanine and cytosine units, as well as adenine and thymine units, were the same in double-stranded DNA. During the late 1940s, Barbara McClintock discovered the mobility of genes. Her discovery of the jumping gene, or the idea that genes can move on a chromosome, earned her the Nobel Prize in Physiology. In 1951, Rosalind Franklin's work in X-ray crystallography began when she started taking X-ray diffraction photographs of DNA. Her images showed the helical form, which was confirmed by Watson and Crick nearly 2 years later. And finally, Watson and Crick published on DNA's double helix structure that twists to form the ladder-like structure we think of when we picture DNA.
Lastly, we are going to mention four scientific advantages. First, we have sequences. The primary purpose of sequencing one's genome is to obtain information of medical value for future care. Genomic sequencing can provide information on genetic variations that can lead to disease or can increase the risk of disease development, even in asymptomatic people. Second, we have genome editing. Through a genome editing procedure called CRISP, scientists can modify aberrant genes. Gene editing techniques use enzymes called nucleases to snip DNA at specific points and then delete or rewrite the genetic information at those locations.
Third, non-invasive prenatal tests. Advances in DNA sequencing make it possible for us to detect genetic abnormalities in a fetus with only a blood sample from the mother through non-invasive prenatal screening, which is a screening method that allows us to identify those fetuses that have a high risk of presenting genetic problems. Fourth, cloning, which is replicating a fragment of DNA placed in an organism so that there is enough to analyze from or use in protein production, first began in the 70s when cellular codes were being investigated and manipulated. And in 1996, a mammal was cloned for the first time from an adult cell.
Thank you for your attention.